All right, our chapter today is about mitosis and meiosis. Mitosis is happening on the left, meiosis is happening on the right. We will talk about the details of each as we go through. So cell reproduction. Reproduction, replication, they're all kind of used interchangeably, but they're processes for each. So reproduction can mean either the birth of brand new organisms, but more commonly involves the production of new cells. Your body is constantly making new cells, especially stuff like your skin cells. Your skin cells kind of get scraped off every single day, and your body has to make new ones. So those cells reproduce in order to make new skin cells, so your skin's not all exposed to the elements. So when a single cell undergoes reproduction or cell division, two daughter cells are produced that are genetically identical to each other and to the parent cell. Now, I don't know why we go with daughter, we go with sister. Scientists just once upon a time said we're going to do that instead of son and father. So we refer to when a cell replicates itself, ends up with two cells, we refer to the offspring as daughter cells. Before a parent cell splits in two, it duplicates its chromosomes, the structures that contain most of the cell's DNA. During cell division, each daughter cell receives one identical set of chromosomes from the lone original parent cell. Cell division plays important roles in the lives of organisms. Like I said, it replaces damaged or lost cells. It also permits growth and allows for reproduction. Some organisms do asexual reproduction, some do sexual reproduction. Our bodies do both. When we need to replace cells, our cells do asexual reproduction. They just clone each other and split in half. But when we want to actually have babies, our bodies do sexual reproduction. Good morning, Daniela. Good morning, Christian. Asexual reproduction are single-celled organisms reproduced by simple cell division. There's no fertilization of an egg by a sperm like we do. Some multicellular organisms, such as sea stars, can grow new individuals from fragmented pieces. They do this by asexually reproducing their cells. Also, growing a new plant from a clipping is another example of asexual reproduction. Another word for asexual reproduction would just be cloning. These organisms clone their cells, so all the cells are exactly the same. They have the exact same DNA and information. These are some examples. Bacteria reproduce asexually. They just split in half and clone themselves. Regeneration of a salamander limb. So lizards, a lot of reptiles, you cut off a limb, a tail, a leg, a toe. They will regenerate those pieces. And then starfish do it too. That's actually one of the more common examples. If you cut off the arm of a starfish, that starfish will grow a new arm, but that arm will also grow a new starfish, like in the picture. And then plants, a lot of the times, you clip a piece of the plant and you bury it in dirt, it's going to come out with the same exact plant. Asexual reproduction conveys an advantage when plants are sparsely distributed and unlikely to be able to exchange pollen. or superbly suited to a stable environment. So there's no need to exchange sperm and eggs or pollen in this case, in plant cases. If they're already extremely suited to their environment, they can just clone themselves and live on forever. Asexual reproduction also eliminates the need to expend energy. Organisms that reproduce this way, they don't waste energy forming gametes, the eggs and the sperm. They don't waste energy copulating with a partner. In nature, things do not waste energy when they do not need to be done. In asexual reproduction, the lone parent and its offspring have identical genes. And mitosis is the type of cell division responsible for both asexual reproduction and growth and maintenance of multicellular organisms. So 99% of our cells perform mitosis to be able to repair and grow, duplicate themselves when needed. Our tissues and cells only last so long. Our body's constantly replacing them. Our skin cells, like I said, is the prime example. We're constantly shedding our skin cells. If you have dust in your house, most likely it's a lot of skin cells. And our body's constantly remaking them. The opposite end is sexual reproduction, requires fertilization of an egg by a sperm, which are made by a special type of cell division called meiosis. That'll be the second half of today's notes. So like I said, 99% of our body cells perform mitosis when they just need to heal or make new cells. But we also make sperm and eggs, depending on if you're a boy or girl, obviously. And those are made by meiosis. And thus, sexually reproducing organisms use meiosis for reproduction and mitosis for growth and maintenance. Sexual reproduction has an advantage when speeding adaptation to a changing environment or allowing a population to more easily rid itself of harmful genes. Normally, when an organism has harmful genes, it does not get to reproduce. So that gene just dies out with that organism. They can't attract a mate, they can't reproduce, they don't have the right body parts, they don't have the right cells. Nature finds a way to make sure that organisms with detrimental genes don't pass on those genes to offspring. So to review, you guys already went over this in chapter 4, I think. In a eukaryotic cell, most genes are located on chromosomes in the nucleus. A few genes are found in the mitochondria and chloroplasts, but most of it is in the nucleus. Each one contains one very long DNA molecule, typically bearing thousands of genes, each chromosome. And the number of chromosomes in a eukaryotic cell depends on the species. There's a good picture of the organization of chromosomes. Get unwound all the way to DNA, all the way to the nucleotides, the ACTG, which we'll go over in the next chapter. Number of chromosomes in body cells. So like I said, different animals, different plants, different organisms have a different number of chromosomes. Humans, right in the middle, we have 46 on average. A mouse has 40, a giraffe has 30, and so on and so forth. Chromosomes are made of chromatin. Fiber is composed of roughly equal amounts of DNA and protein molecules, but they are not visible until cell division occurs. They mostly just look like kind of blob, shaded in blob in the middle of a cell until cell division starts. That's okay, Shay. I'll post the video later for whatever you missed. So to the left, over here is what cell normally looks like. That's the nucleus, but it's all shaded in. You can't really see anything. Once cell division starts, now you can see these little squiggly things. These are when chromosomes really start to form at the beginning of mitosis and meiosis. The DNA in a cell is packed into an elaborate multi-level system of coiling and folding. Histones are proteins used to package DNA in the eukaryotes. And then nucleosomes consist of DNA wound around histone molecules. This is just different vocabulary for different pieces of chromosomes found in the nucleus of your cells. There's a picture of it. You got the DNA double helix. You can see it winds around the histones, the proteins. And then the protein and the DNA together are called nucleosomes. And they look like beads on a string if you look at them under an actual microscope. Those tighten even further and coil and twist around until it forms that X-shaped structure we know as a chromosome. When it looks like an X, it's already been duplicated. Each half is called a sister chromatid, and the middle part is called a centromere, which are important vocabulary words as we get into the process of mitosis and meiosis. So before a cell divides, it duplicates all of its chromosomes, resulting in 2 copies called sister chromatids. And each contains identical genes. 2 sister chromatids are joined together at a narrow waist called the centromere. So this is just a general example. That's 1 chromosome, it duplicates. Now you have that X-shaped structure with the sister chromatids. Eventually, those are going to split off and distribute to the daughter cells. So when the cell divides, the sister chromatids of a duplicated chromosome separate from each other. Once they are separated, each chromatid is considered a full-fledged chromosome and identical to the original. So just because it splits in half doesn't mean the information splits in half. The information is still the full 46 that gets passed on to a daughter cell. The cell cycle is the ordered sequence of events that extend from a time a cell is first formed from a dividing parent cell to its own division into 2 cells. And there's 2 distinct phases in the cell cycle, the interphase, which the cell spends about 90% of its time in, and then the mitotic phase, when mitosis happens. The G1S and G2 phases of the cell cycle are grouped together and called interphase. Most of the cell's life is spent in interphase. You can just tell from the graph. During this phase, the cell performs normal functions and prepares to divide by growing and replicating its DNA, as well as everything else in its cytoplasm. All the organelles, everything gets duplicated in preparation to pass it on during mitosis. So interphase under a microscope is going to look like the cells on the right. They're just all shaded in. Maybe you can see little dots here and there. But in your lab this week and on your test, on your practice questions, you're going to be looking at different phases under a microscope. So you have to be able to pick them out. So those cells are all in interphase. You can see different cells in different phases in that square, but we're going through the phases at the moment. So interphase is just a big blob all shaded in. Mitosis and cytokinesis is also called the mitotic phase. It divides the cell's nucleus and cytoplasm in 4 phases and produces genetically identical daughter cells. This is what our cells do to maintain and grow and repair themselves when we need it. Prophase, chromosomes in the nucleus condense or get thicker under the microscope. You can actually see strings forming. Let's see below, that should say below left. Centrioles and spindle fibers form a kind of net, and the nuclear membrane starts to disappear. That should say, let's see, below left. So on the left, that's what it looks like under the microscope. Those are the ones that you will have to identify in your quiz, in your lab, in your practice questions. And then on the right, it's just kind of an animation of what's happening. The nuclear membrane disappears, chromosomes condense, and you can actually see chromosomes. Metaphase, chromosomes line up individually in the middle of the cell. This is called the metaphase plate. The net spreads out and catches the chromosomes at the centromere. That net are the spindle fibers. And like on the right, there's an animation and it looks pretty. Chromosomes are all lined up and you can see the X-shaped structure. But on the left, that's what it actually looks like under a microscope. And you can even kind of see the spindle fibers, the grayish net that's kind of catching them. Anaphase, sister chromatids separate to opposite sides of the cell, and they're being pulled apart. So they're in metaphase and they're being pulled apart by the spindle fibers. And again, on the left, that is a cell that you will have to identify eventually in tests and quizzes and labs. telophase, the nuclear membrane reforms. Chromosomes begin to uncoil, and the cytoplasm kind of stretches out and begins to separate. So it looks like two little nuclei inside a single cell. So it goes from metaphase to anaphase. They're still kind of separated, but telophase, they just kind of start to tighten up again. Cytokinesis. Cyto, we've learned before, means cell, and kinesis means split. So it's the actual splitting of the cell and its cytoplasm in two. In animal cells, a membrane forms a cleavage furrow and kind of just pinches closed. That's what's happening on the top animation. In plant cells, a cell plate forms, and that cell plate eventually becomes the new cell wall. So in plant cells, cytokinesis begins when vesicles containing cell wall material collect at the middle of the cell and then fuse, forming a membranous disc called the cell plate. And then the video is the crash course video about mitosis that you guys can watch if you want. You'll be watching the meiosis one for your Edpuzzle this week. Cancer cells growing out of control. We should remember this from last semester. We went over cancer a lot last semester. So normal plant and animal cells have a cell cycle control system that consists of specialized proteins, which send stop and go ahead signals at certain key points during the cell cycle. So all these checkpoints, checkpoint 1, checkpoint 2, give the cell the green light to keep replicating or red light to stop, something's wrong. Cancer is a disease of the cell cycle. Cancer cells do not respond normally to the cell cycle control system. They see a red light, they just keep going. They see a green light, sometimes they stop. Cancer cells can form tumors, abnormally growing masses of body cells. If the abnormal cells remain at the original site, the lump is called a benign tumor. The spread of cancer cells beyond their original site of origin is called metastasis. And this is when they become malignant tumors. They spread to other parts in the body and they interrupt normal body functions. And a person with a malignant tumor is said to have cancer. Meiosis is the next process we'll go over. This is involved in sexual reproduction. Sexual reproduction depends on meiosis, which is the process that makes eggs and sperm, and then the fertilization, the coming together of those two to make a new offspring. And the offspring produced from this process contain a unique combination of genes from parents. This process is the reason why you and your brothers and sisters, even if you have the same parents, you still don't look exactly alike. Because meiosis and fertilization make sure you have unique genes. Homologous chromosomes. So we know what chromosomes are, but homologous chromosomes are two chromosomes with the same structure. Homologous is a big science word, but homo means same, logos means structure. So they're just two chromosomes with the same structure. Different individuals of a single species have the same number and types of chromosomes. A human somatic or body cell has 23 pairs of chromosomes, 46 total. And these are the cells that go through mitosis to replicate. Alleles. Homologous chromosomes can possess different versions of the same alleles, or of the same genes called alleles. Eye color is always a typical one. Eye color is located on a certain place in your gene, but not everybody has the same gene for eye color. Some people have the gene for brown eyes. Some people have the gene for blue eyes. Some people have the gene for green eyes. The different versions are called alleles. And you get one allele from each parent. Like I said, we have 23 pairs of chromosomes in all of our somatic or body cells. The fact that they're set up in pairs means you get one from each parent, one from mom, one from dad. A karyotype is an image that reveals an orderly arrangement of homologous chromosomes. You can see in there, there's 22 pairs, and then the last pair, the 23rd pair, is a little bit different. Those are the sex chromosomes that will typically reveal gender. And this is how we get the chromosomes. Actually, I'm not going to watch that right now because that's something we watched already. The amniocentesis, when they stick a needle in the mom's stomach and pull out the cells from the baby surrounding the baby, and then they put it in this picture and determine if they have exactly 46 or if they have one missing or one extra. But humans have 22 pairs of matching chromosomes called autosomes, and then the 23rd pair called the sex chromosomes. Girls are supposed to be XX, boys are XY, but we know there are variations to this. Sometimes there's 3 X's, sometimes there's XXY. The life cycle of a multicellular organism is the sequence of stages leading from the adults of one generation to the adults of the next generation. Humans are called diploid organisms, which means their body cells contain 2 sets of chromosomes. However, their gametes are called haploid because they only have 1 set. So if you think haploid has half the information, and the gametes are the egg and sperm cells. So our body cells, our somatic cells, are called diploid because they have the full 46. Gametes, the eggs and sperm, are called haploid because they only have 23. Sexual life cycles involve an alternation of diploid and haploid stages. In humans, a haploid sperm fuses with a haploid egg during fertilization to form a diploid zygote. Meiosis is the process that forms these haploid gametes, the eggs and sperm. And this process keeps the chromosome number from doubling every generation. So our body found a way to cut our sex cells or our gametes in half so that the next generation, when we eventually have offspring, has the correct number. We can't each pass on 46, then the next generation would have 92. And that's bad. So our bodies cut this number in half for us, so that each subsequent generation has the correct amount. An overview of meiosis. Interphase is followed by 2 consecutive cell divisions. All chromosomes are duplicated during interphase. The first division, the round is just called meiosis 1, segregates 2 whole chromosomes of a homologous pair. And the second division, meiosis 2, separates the sister chromatids. And this time when the sister chromatids get separated, the information also gets split in half. So this is, again, an overview of meiosis. What happens during interphase before meiosis, the pair of homologous chromosomes in a diploid parent cell line up together, then they duplicate. During meiosis 1, the homologous chromosomes separate. And then during meiosis 2, the sister chromatids separate. And in the end, when the sister chromatids separate, you're left with half the information in each cell. So they each have the same phase names. You have interphase and then prophase 1, metaphase 1, anaphase 1. During anaphase 1, there's some stuff that's highlighted that's important. Sister chromatids remain attached, and just the pairs of homologous chromosomes split up. Telophase 1 and cytokinesis is not shown in here because it's the same. Crossing over is a very important process that happens in prophase 1. It's the exchange of chromosome segments between homologous chromosomes. And it results in new combinations of chromosomes, which leads to increased genetic diversity. It's also known as genetic recombination. So you can see on the bottom, you have a purple chromosome and a green chromosome, but they have the same structure. So this is a pair of homologous chromosomes. They line up next to each other. In the middle panel, you see the legs just kind of cross over. And then in the end, on the panel on the right, they switched information. Now the purple chromosome has a little green leg, and the green chromosome has a little purple leg. And again, this is just another overview. And this is round 2, prophase 2, metaphase 2, anaphase 2, and it's highlighted, the difference. Sister chromatids separate. And then telophase 2 and cytokinesis. You end up with 4 haploid daughter cells. So when we compare the two, in both processes, the chromosomes duplicate once during the preceding interphase, but the number of cell divisions vary. Mitosis uses 1 cell division and produces 2 diploid cells. Meiosis uses 2 divisions and produces 4 haploid cells. And all the events unique to meiosis, like crossing over, occur during meiosis 1. So this is just a side-by-side comparison. Main differences are highlighted. Offspring of sexual reproduction are genetically different from their parents and one another because of the crossing over process. This is another reason that you are not identical to your brothers and sisters, even though you have the same parents, because of this crossing over process. And the GIF is meiosis in a nutshell, crossing over and all. And you can see the ending, the last 4 gametes are all unique. They all have different combinations of that green and yellow. Independent assortment. When aligned during metaphase 1, the side-by-side orientation of each homologous pair is a matter of chance. It is random. There is no way to predict it. There is no way to cause a certain combination. Every chromosome pair orients independently of all the others at metaphase 1. As long as they have the same structure, they're colored a certain way, just so you can see it, but they each have the same structure and they line up together. And then random fertilization. At the end of meiosis, you have 4 haploid gametes. A random human egg cell is fertilized by 1 random sperm, leading to genetic variety in the zygote. Remember, the zygote is what the baby is called when it first just becomes a clump of cells. Then it turns into an embryo, then it turns into a baby. If each gamete represents one of 8,388,608 different chromosome combinations at fertilization, humans would have twice that, or that squared, more than 70 trillion different possible chromosome combinations. What happens when errors occur in meiosis? These errors are called non-disjunction. It happens when the members of a chromosome pair fail to separate at anaphase, which produce gametes with an incorrect number of chromosomes. Non-disjunction can occur in either round, 1 or 2. And such mistakes can result in genetic abnormalities that range from mild to fatal. So in meiosis 1, homologous chromosomes can fail to separate, so that in meiosis 2, the sister chromatids could fail to separate, and then the gametes end up off. You can see the abnormal, if it fails to separate in meiosis 1, all 4 gametes in the end will be abnormal. If it fails to separate at meiosis 2, the sister chromatids, then you got a 50% chance of passing on a normal egg or sperm, and then a 50% chance of passing on an abnormal gamete. If an organism survives, it will have an abnormal karyotype, usually with either 1 extra or 1 less chromosome, 47 or 45. And that child will end up with a syndrome of disorders caused by the abnormal number of genes. So abnormal karyotypes, there's some examples here. The one on the left has an extra X. That's called Klinefelter syndrome when they have an XXY. On the right, they only have 1 X. They only have 1 sex chromosome. So they have 45 altogether. Now it's called Turner syndrome when girls only have 1 X. Couple more. On the left, that person has trisomy 18. That's called Edwards syndrome. And then on the right, that person has a karyotype of 21. And they have a 3rd chromosome in the number 21 spot. That is Down syndrome. That's one of the most common. Down syndrome is also called trisomy 21 because they have 3 copies of chromosome 21 instead of the standard 2. It affects about 1 out of every 700 children. And the incidence of Down syndrome increases greatly with the age of the mother. So as a woman gets older, if she gets pregnant when she gets older, they always recommend an amniocentesis so the mother can be prepared just in case there's any of these disorders. And this is just a graph. Infants with Down syndrome and then the age of the mother, you can see it goes up exponentially after the age of 35. And this is for you guys to quiz yourself. This is what you're going to see on your quiz and your test and your lab this week and next week. So make sure you know how to identify all of these cells that you would see under a microscope.